Thank you, Dr. Alan Chambers of the University of Florida for sharing to us this presentation about vanilla genomics, diversity, and horticulture.
All right, thank you, and it's a pleasure to give this presentation. Always excited to reach out to other people that are interested in orchids, especially vanilla.
Thank you, Dr. Alan. Let's start.
Okay, so just a quick introduction. So where we are located, I am at the University of Florida and the Tropical Research and Education Center. For those of you who are aware of the United States, so this is Florida, shown here is our main campus, which is about 400 miles north of us, so we're right near Miami. Our research center was established in 1929 in Homestead. We have 160 acres and 17 faculty covering multiple disciplines from plant pathology to breeding, to entomology, to soils. And in general, our job is to support the $1 billion local agricultural economy, as well as to conserve natural resources for future generations. Our area here is unique in the United States. We are a subtropical climate. So we have both distinct rainy and dry seasons, as shown here, with rainfall around 5 centimeters in the winter months, and up to more than 20 centimeters per month in the rainy season, which we're in now. Our average temperatures are warm and beautiful. We do get freeze events every few years where at night the temperature might drop to 30 C for a few hours, but because it's not very long in duration, we can grow many tropical crops here, but there are still some that we cannot grow. My program is brand new. It started 4 years ago. My job is to produce new cultivars that have pest resistance, stress tolerance, and better fruit quality. I spend 80% of my time doing research and 20% with extension. I work on any number of different tropical species depending on the needs of the growers. And some of those are shown here, with mango and vanilla being some of my primary species. This is a picture taken from an airplane flying into Miami. Our area is unique for most of Florida in that we have very little land for agriculture. So shown in this image at the top is preserved land. This is the Everglades. And then towards the bottom of the picture is the Atlantic Ocean. And this is one area that's entirely been filled with housing. South of here, the land opens up a little bit, but land prices are extremely high because of the number of people that want to live in this environment, this subtropical environment. And as a result, our growers have to rely on highly profitable crops in order to stay in business. And that's where my job comes in. So where did vanilla come from? Vanilla originates from Mexico and Central America. So as the legend goes, there was a princess born that was too beautiful to be given to any mortal in marriage. Her name was Morning Star. So she was given to the goddess of crops and sustenance, where she worshipped in the temple each day. As she was working in the temple, a young prince named Young Deer fell madly in love with Morning Star, and he would sit and watch her as she went to the temple and back each day. One day his passion overcame him, and he jumped out of his place where he was hiding in the bushes. He grabbed Morning Star and ran with her off into the mountains. As they were running, Morning Star fell in love with Young Deer as well, and so they together decided that they would escape. As they were running through the mountains, they were turned back by a fire-breathing dragon. Back to the main road, they were then captured by the priests of the god of crops and sustenance, where Young Deer, the prince, was killed on the spot, followed by Morning Star. And their hearts were cut out and taken back to the temple to offer a sacrifice for Morning Star's infidelity. But from the spot where their blood was spilt grew a bush, and on that bush an emerald vine that produced beans of the sweetest sustenance. And that's where we get vanilla from. So starting in the 1500s to 1600s, vanilla migrated from its home in Mexico and Central America. It wasn't until, so it first spread into the Caribbean islands, and then to Hawaii, the Philippines, and then through Indonesia and surrounding countries. It wasn't until the 1800s where botanists and hobbyists were able to get the vanilla vine to successfully grow in European greenhouses. There were a lot of unknowns, but a lot of interest in botany at that time. From Europe, the vines traveled through colonies down to Madagascar and the islands surrounding. And it's descendants of those original clones that now make up the production in Madagascar today. The United States is the largest importer of vanilla beans in the world, followed by the European Union. The vines are mostly grown, as shown here, in this type of jungle agroforestry system at about 1,000 vines per acre, or just more than that per hectare. The vines, because of their high profitability, are subject to theft, especially these last few years. And so the growers will actually stay in the field to protect their crop. These vines or the beans are harvested, they're cured in Madagascar, and then they're shipped to the United States, where they're then cured into the vanilla extract that's then used in cooking, perfumes, cosmetics, by homeowners as well as major food and beverage companies. There is a vanilla standard of identity, so you can only legally sell Vanilla planifolia and Vanilla tahitensis as two species as vanilla extract. And this shown here, as vanilla extract prices increased, what we've seen is more blending of vanilla. So natural vanillin comes from vanilla beans, and artificial vanilla comes from a number of other different sources, but because it's so much cheaper, we're seeing it blended into the natural stuff. Vanillin is the world's most popular flavor. It's used in a number of products, including being used to mask the bitterness in chocolate. Only 1% of the vanilla flavor in the world actually comes from vanilla beans. Artificial vanillin mostly comes from waste products of the paper and pulp mill industry. And there's a quality difference between just the one aroma compound, vanillin, versus vanilla extract, which has vanillin plus also dozens of other compounds that give it a rich and full flavor. Because of challenges in production in Madagascar producing 80% or so of the world's total vanilla beans, whenever there's a major weather event or geopolitical instability, the prices of vanilla skyrocket. And so as a result, there's dwindling supply. There's never enough supply of the beans. And in recent years, a number of major food and beverage companies have pledged to remove artificial ingredients from their product offerings. So as a result, there's more demand for vanilla beans on an already stressed market. So we're interested in expanding cultivation as a new economic opportunity for South Florida growers. Those challenges even reach down into Florida. So our local businesses are impacted by vanilla prices. It's usually the #1 most expensive ingredient, whether you're making cosmetics or you're a bakery. And these challenges have led us to start asking why we don't produce vanilla in new geographies. So what does it take to grow vanilla? Vanilla is primarily propagated by cuttings, usually 30 to 100 centimeters long. So this is clonal propagation, which means that each plant is genetically identical to the parent. This also means that there are problems with viruses. So if the maternal plant has virus, then most likely the daughter plant will have virus as well. There's also problems with unknown genetics. A number of people are growing what they think is Vanilla planifolia, but it's actually not, and that has legal complications. You can grow the seeds in vitro, but they, like siblings in a family, each of those seeds, those plants will be different than the parent, sometimes better, sometimes worse. But that's also an opportunity for us to create new and better types of vanilla. Shown here are some different types that we've seen in tissue culture. These each have the same parent, but looking at the different leaf sizes and shapes, as well as the abundance of these aerial roots, whether that's stable or heritable or not, we're still investigating that. Vanilla is not a picky plant to grow. I would say that it does require shade, so it's a shade-loving plant. In shade houses like this shown here, we use about 50% shade for the vanilla vines. Vanilla really is a wild species that we've cultivated, so it hasn't really been domesticated, and it has a number of those wild traits in it. But as a benefit of that is that it doesn't have strict requirements for substrate, and so we use just a standard mulch for the terranean roots. And then vanilla is an orchid, but it's a climbing orchid. And so we'll let it climb up about 6 feet, and then it will loop back down to the soil where it puts out new terrestrial or feeder roots before climbing back up again a piece of bamboo or a tree. Shown here are alternative production zones or areas where you can grow vanilla underneath existing, say coconut plantings, avocado trees, or is this type of agroforestry system in more of a wild system, you could produce vanilla and another crop. In Madagascar, what does the production look like? So I was fortunate to have funding to go to Madagascar and see commercial production there. Again, vanilla is grown in partial shade. If the growers are more attentive to the vines, they will put mulch down, but it still requires a trellis. The planting, they transfer cuttings from mature vines and put those in new tutor trees here. As you can see, this man has put in new tutor trees. This is a local tree that grows well in Madagascar, and they'll put one vine on each tree with a little bit of straw here to try and keep the weeds down, though they don't do much weed control. After about 3 years, the vines are mature, and then they start flowering. Now in Madagascar, they don't have any natural pollinators, and so each vanilla flower has to be manually pollinated. That is not the case in Mexico and Central America. I'm not sure down about in Colombia, but even in Florida, we do have natural pollinators that will find the flowers and pollinate them for us. Though we're not sure exactly what those insects are, we're actively conducting research to identify those, because a commercial industry that doesn't have to manually pollinate flowers would save about 40% of their input costs. After pollination, the beans take about 7 to 9 months to develop. The longer the beans are on the vine, the better aroma that they will have. And unfortunately, because of theft, the growers are left with decisions of harvesting early or letting most of their beans get stolen. So either theft is leading to premature bean harvest, or the growers themselves are trying to keep something of their harvest, and they harvest early, and you end up getting a lot of beans that just don't have the quality that you would expect. For many of you, think about harvesting a mango or any number of other tropical fruits. If you harvest them too soon, they just don't have that quality. They're not as sweet, they're not as flavorful. The same is true for vanilla. The beans are harvested, where then they're cured, which is a series of thermal treatments, so heat treatments over time. A green, fresh vanilla bean does not actually have any aroma, but it takes this curing process to bring out the aroma. Then those beans are shipped, where they are extracted and then sold as extract. Shown here are 2 different kinds of vanilla. This is Vanilla planifolia and Vanilla pompona. These grow somewhat side by side in Madagascar, though the pompona types you cannot legally sell as vanilla extract because it's the wrong species. Again, you can only sell Vanilla planifolia and tahitensis out of the 110 vanilla species. Those are the only 2 you can sell as vanilla extract. This is a couple of beans that are left on a grate here. So as part of the slow dry down process, they'll put the beans on these grates, take them outside and expose them to the sun for a few hours a day, and do that for a few months as part of the curing process. And then these beans are shipped to the major importing countries, again, United States and the EU. So what does it actually look like to pollinate a vanilla flower? This is Vanilla pompona. It has a yellow flower. And the major parts of the flower here, this is the pollinia. If you know orchids, this is where the pollen is. This is a flap-like structure called a rostellum that prevents under natural conditions, the pollen from fertilizing the stigmata, which are found here. So this has to be circumvented during natural pollination by lifting the raw stellum and then lightly touching the pollinia to the stigmatic surfaces. Vanilla in Florida. So we've been working to establish a new industry of growing vanilla in Florida. We have had Vanilla planifolia, the commercial species, in South Florida since at least 1910. Those are the earliest records we have as part of its migration through the Caribbean islands, or maybe a remigration, because we know vanilla moved much earlier than that through the Caribbean. Vanilla planifolia has this cream green flower. It's a good climber, and so this is shown in a more natural ecosystem where it's climbing up the trees, has a zigzag pattern from leaf to leaf. These are beans that were naturally pollinated in Florida, but we don't know what insect is actually doing the pollination. And then shown here is another Vanilla planifolia from just another side. Vanilla pompona has a characteristic yellow flower. It's a very vigorous plant. It's very strong. It has larger leaves and a larger stem. It's growing up a palm tree here, which is tolerating heavy exposure to full sun. And this is a giant oak tree. This is a backhoe or a tractor for comparison to size for how big this vine is. We have 4 native species in South Florida. This is Vanilla phayantha, has a beautiful white corolla flower. This is in a sluice, so the water level actually rises to about waist high during some seasons of the year. And these are naturally occurring pods of Vanilla phayantha. This is Vanilla mexicana, has a beautiful white labellum, modified lower petal. A beautiful specimen I found here. And again, this also produces beans without any manual intervention. Our native vanilla species are endangered. I'm sure it's very similar to Colombia's native species. And they're very threatened as well. So I had this site that I had research permits to go and investigate. And we had a hurricane come through, and I visited the site 2 weeks later. And this very beautiful specimen, which is the largest I've ever seen, a palm tree fell on it and killed this one plant. And you do that 100 times and you've wiped out the population. We have 2 leafless species, Vanilla barbellata. This has more red to the modified lower petal. It does not produce leaves, as you can see here. It has a sprawling habit, and the flowers are very aromatic. And Vanilla deloniana no longer exists in the wild because of illegal overcollection. But it does exist in private collections, and we have a permit to legally own and propagate this plant. So vanilla diversity, as I said before, there's around 110 species of vanilla. In Mexico, Central America, the Caribbean, and Florida, we have 14 species. And then down in northern part of South America, around 54 species, so a lot of diversity down there. As well as through parts of Africa, there are even native species in Madagascar, and then through Indonesia and surrounding islands as well. When we used to look at diversity, we would do single gene sequencing. So you would take one gene, usually this is a maternally inherited gene, so a plastid sequence that you only get from your mom. So it tells you who your mom was, and we would spend maybe $5 and get a sequence of 800 base pairs, and maybe 5 of those base pairs would be what we call a single nucleotide polymorphism, or a SNP. Those are shown here. So this is a sequence of DNA between different species of vanilla. And anytime you see a colored box, that's where one DNA nucleotide was different from everyone else. And we call that a SNP. So for example, this Vanilla planifolia has a T instead of the more common C. And we can use that as a type of DNA fingerprint to understand how different plants are related or how they are different. So we can take these different species and we create what you can consider as a family tree. This is a phylogenetic tree using this DNA information. Those plants that are more similar are on the same branches of the tree, and those that are different are on separate branches of the tree. So as you can see here in the blue, Vanilla planifolia are all on the same branch, versus Vanilla pompona is on its own branch. This is good, but it has very poor resolution. So for example, if you have a hybrid plant between Vanilla planifolia and its daddy was Vanilla pompona, it would show up as planifolia because you can only detect who the mother was. So we actually have a number of hybrids of vanilla which were going unnoticed, undetected because of these former methods. There were also sterile types that we couldn't identify. There were a lot of sterile vanilla plants out there. You can grow them for years and pollinate the flowers with all of the money and time it takes to do that and never get a bean off of those plants. There are also a number of plants where there's no flowers to quickly identify the species, and yet growers want to know what it is. And that can be very challenging if you don't have that DNA fingerprint in your database. So we designed a new study because diversity is very useful for genetics in plant breeding. The study had 112 accessions and including over 23 species. And that was over a year ago, and since that time we've continued to grow our vanilla collection. We generated a draft genome for vanilla planifolia, and we sequenced around 18 billion DNA bases and found thousands of these SNPs that we could use. Again, a SNP are these individual DNA changes that we can use to figure out how different plants are from others. This is a picture of our collection. We grow them on individual trellises at about 1 meter square spacing per plant. This is a list of the different species in our collection. Mostly we had Vanilla planifolia because that's the commercial species, and Vanilla pompona, and what we thought were Vanilla tahitensis, which is the other commercial species. So whereas before we could only generally tell differences based on the simple sequences, now we're using 5,000 of these individual SNPs to make a very nice DNA fingerprint. Shown here in light blue are the Vanilla planifolia plants. If this is a structure plot, so it takes blocks of DNA and assigns a color to them. So if, for example, this light blue, these are blocks of DNA that are most commonly associated with Vanilla planifolia. So if you've got a long blue stripe, you're Vanilla planifolia. Versus, say, a long green stripe, which is more common in pompona. Then you can start to identify hybrids here. This plant here looks like it was an original cross between a planifolia and a pompona. Then someone took pollen from that hybrid and then put it onto a flower of Vanilla planifolia again. So it's 1/4 pompona and 3/4 planifolia. And the only way you can identify that is with the methods that we've been developing. We also were able to identify other hybrids, say with Vanilla phayantha and odorata, and then a number of native species. And this helps us to understand how distantly related plants are. The more data, the better in this case. So whereas Vanilla planifolia, we might consider how those would be your family, your cousins, then your distant relatives. The farther you go, these are people that are your neighbors or people you might meet in a taxi ride or an Uber pool, then maybe people that you'll never meet in person, followed by real diversity, just crazy different plants that we don't even know if they're hybridizable with Vanilla planifolia. What does this mean for us in breeding? We need to stay with Vanilla planifolia and tahitensis to make sure that something is commercially viable. Otherwise, we have to work with changing regulatory status and laws. But even within here, we can look for things like how many beans do you get per plant? We want beans that don't split on the vine because those are less valuable. Or disease resistance, like resistance to fusarium. If we go farther away, we have no idea what we can find. So maybe again, disease resistance, plants that grow faster or they're stronger, maybe plants that pollinate themselves, maybe different flavor compounds. And then distantly related, we have no idea what we're going to find until we actually look at it. The nice thing about investing in those big data sets is that then we can use that information to look for cheaper ways to genotype our material. So for example, we took some of those SNPs that were common in Vanilla planifolia or Vanilla pompona, and then we use those to characterize a population again. But instead of taking 3 months to 6 months to get our data, you can actually use this type of information in 1 day to go from leaf to your data and get your results for maybe a few dollars or a few cents, depending on how many samples you're running. What that looks like here, this is using a quantitative real-time PCR machine. We found a piece of DNA that was only present in Vanilla planifolia and not present in Vanilla pompona. So the higher the number here, the less of that there is in the sample we ran. Then we tested it against vanilla planifolia hybrids, self-vanilla planifolia, and then planifolia pompona hybrids and pompona planifolia hybrids. So this confirms that, especially these pompona planifolia hybrids, that they do have planifolia in their background. And then we can do that for a number of different targets. Here we can do the exact opposite thing for vanilla pompona. We found a sequence that was present in vanilla pompona that was not present in vanilla planifolia. It was not present in the planifolia self progeny, but it was present in the hybrids with vanilla pompona, except for this individual here. And then we can take those same DNA tests and we can run them against all of the species in our collection to see how unique they are. And now we can do this faster and we can do it cheaper to identify hybrids. So for example, we had unknown accessions that showed up with both vanilla planifolia and phantha DNA fingerprints. So we know it's a hybrid between those two species. And in that case, we would not recommend it for commercial production. And then we can start looking at high value traits. This is a PCA plot where we found that 11% of the plants in our collection were misclassified. So they had the wrong species assignment. And then we also found that 10% of our collection were hybrids that we had previously unknown. What we're going to use this information is to look for pods that don't split. So shown here, the pod split. This is a wild trait in many cereals. Vanilla tahitensis, you can leave those pods on the vine and they don't split, and they further develop their aroma. We're very interested in better tasting vanilla beans. So we know we've got something great as a wild species, but maybe with a little bit of effort, we can make it taste even better. Shown here is a flavor wheel of the different components of flavor for vanilla, maybe the caramel notes, the anise notes, woody, fruity, spicy. These are all things that we can work with at the genetic level for new and better types of vanilla. And then shown here, we do GCMS to analyze the aroma. If you were to take one vanilla bean extract, which has hundreds of different aroma compounds, this technique allows us to spread those individual aroma compounds out so that we can identify individual compounds and then quantify them. This large peak here is actually vanillin, and you can see the higher the peak, the more the vanillin you have in your sample. So this is vanilla planifolia, and this is vanilla pompona, and what we can see is pompona just has less vanillin than vanilla planifolia. Since we started this project 4 years ago, our work has expanded to collaborating with scientists in over 15 countries, shown here in blue. We've been analyzing diversity from Mexico to Brazil, and this is exciting because there are many secrets that we're uncovering. Some species are actually not distinct from other species. Some species are simply hybrids between other species and shouldn't have their own species designation. We're also digging into some of the history of where vanilla came from and how it evolved, which is extremely exciting. And the reason it's so nice with the collaboration is that there's things that we can't do by ourselves. So while our expertise might be in genomics and analysis of genomics information, diversity is found in many, many different countries. So there's a lot we can do by putting together our strengths and working towards common goals of conservation as well as supporting vanilla production back where vanilla originates from. So in conclusion, we've been very busy with a new vanilla program aimed at supporting domestic cultivation of vanilla, as well as developing new genetic tools, resources, and new cultivars that can be released. So we've learned a lot as we've gone, especially how to grow the plants, working on tissue culture protocols to mass propagate the plants, even protoplast isolation if we want to do some gene editing. Our work involves also going out and conserving our natural species, our native species, and grower trials where we distribute vanilla cuttings to growers around the state to see where we can grow vanilla the best, especially if we get away from Miami. Land is much, much cheaper. And what we hope is that in a couple of years, we'll be able to start selling vanilla extract from Florida, locally grown vanilla, to support local growers, as well as provide more options for those looking to purchase vanilla beans. And this work could be similar for Florida as it is for a number of different countries through Central and South America. For any body of work, it takes a lot of teams, a lot of good people working closely together. Many of those are shared here, shown here, as well as my contact information Should anyone want to reach out, please email me. I'm happy to talk about collaborative opportunities for working together, especially because we know that Colombia has great vanilla diversity. And then shown here is a picture out of my window showing the beautiful courtyard where I work. And this alligator was waiting for us one day after we were walking through the slough up with water up to our waists. We came back up out of the slough onto the road, which was slightly under the water as well, and found this alligator just waiting for us, which is a different experience for someone who's trained as a molecular biologist and genomicist, but a nice adventure all the same. And with that, I'll thank you for your time, and again, encourage you to reach out if you have any questions. Thank you.
Thank you, Dr. Chambers.
Yeah, and that's a great question, and the reason is it's historical. So in the early 1900s, the market, vanilla production was struggling. It was mostly focused in Mexico and Central America, mostly in Mexico. And there were a few bad actors that were adulterating vanilla extract. So they were taking their vanilla extract and adding glycerin, glycol, antifreeze, all sorts of different chemicals, and then selling the extract because the beans were very expensive, but these other solutions were very cheap. And so in response to that, they created the rules for what actually is considered vanilla extract. So now there's a legal definition of not only the species you can use, but also how many grams you have to put in the solvent, how much alcohol has to be in the solvent. So it's an alcohol water solvent for extraction. And then that was adopted in the US and in Europe. They have very similar standards. And of course, for us, if people have been consuming vanilla pompona, for example, in Central America for hundreds of years, why should that not be considered vanilla extract? And that is something that can be changed, but currently that's where the limitations are those two species. So around the world, the #1 pathogen people say is Fusarium, but I find some of the data lacking. So we could do a much better job with real diagnostic tests to identify the causal pathogen. So the vines will yellow and die, but that could be from a number of different symptoms. In our own collection, we have had virus, we've detected virus, which is very common in orchids, which will also have the same effect on the vine. The vine will almost, not, I wouldn't say extremely quickly, but it will rapidly, the leaves will brown and the whole vine will die because of the virus. And if you don't start with clean material, that virus can easily spread. We do see caterpillar feeding on a very limited scale, but only for very young plants, very, very young plants. Vanilla leaves produce these calcium rhamphosides that are like microscopic needles. So if an insect comes and eats the leaf, they're also eating these crystal needles, which discourages most insect feeding. Other than that, we don't see a lot of problems because vanilla, again, is a wild species and it's used to fighting for itself, it seems.
Okay. If you say that it's too restrictive to have this commercial potential of the diversity of orchids, vanilla orchids, why did you try to research about all the difference?
#1, as a breeder, I'm intrigued by possibilities. Plus diversity is really what makes my job fun and interesting. Because the legal restriction it's not necessarily science-based, it's more political-based. So if we are able to create something that's wonderful, say a hybrid between vanilla planifolia and pompona, which is actually widely grown in some countries, if the growers want that, the buyers want that because you have better yield, better disease resistance. And if the consumers say, hey, I like this, then the law will change. So we are preparing for that. It says, look, does the consumer say this is vanilla extract? If the consumer says it's vanilla extract, and it's safe, I see no reason why the law shouldn't be changed to reflect that. We still shouldn't dump antifreeze in our vanilla extract, but if it is vanilla and it's a natural product and the consumer understands where it's coming from, then I believe that should be an option, and we're working towards that. But until we have something we can say, look, here's a great example, change your law and you can have it, it's hard to get that argument. So our collection is still very young, and probably only 10% of the collection has actually flowered and fruited so far. But even out of that 10%, we haven't done total yield yet because we need the vines to be mature and robust. But what we're seeing is that there are very big differences in bean length, bean weight, and now we're analyzing extract. So we're seeing beans from maybe 5 to 10 centimeters all the way up to 50 centimeters. And among different species, the bean lengths will change. Longer beans are more desirable. They're worth more money. And then we're also looking at vanilla extract. Are there some types of vanilla planifolia that have higher vanillin content? Or can you feed them differently to get more vanillin? Those are also things we're looking at.
Finally, I get one question, because it's really common to hear the people have some prevention about genomics, treatment of the plants, no? Because they say these cultivars are not natural, they're hybrids. Can you explain why it's important to use the genomic investigation research to improve the cultivars, the crops, and in fact in horticulture and in agriculture in general, not only for our kids?
Well, and I guess it's also important to say that genomics is just a test that we run. It doesn't change the DNA of the plant at all. It just helps us to read the DNA like you would read a book. And we need those tools to make everything more efficient. So imagine if you were trying to bake a cake, but you couldn't see the ingredients. So try to bake a cake with a blindfold on your hands, and all you can do is feel some things. You can do some things well. I know this is an egg, but how can you tell the difference between a flour bag and a bag of sugar, for example? The genomics helps us to answer all of those questions so we know exactly what we're working with. But it also helps us as a tool so that we can work on, say, gene editing. So we can take specific genes and change those. Over the course of human evolution, as we've been domesticating crops as well, we've been changing our plants for tens of thousands of years to make them do what we want. More food per acre, fewer toxins, healthier, more nutritious food. Now what we can do with genomics information is say I find a gene that makes the beans split when they're mature. I can now use my genomics information and gene editing. I can break that gene, and so now the bean no longer splits on the vine, and it will stay on the vine until someone comes and picks it, which will help me save money. So these are tools we're using to take what we have now in agriculture and make it better at a much faster rate. So instead of waiting 1,000 years for nature to do its process, which can be very slow, we are changing things in a single generation. Again, so it takes less land, fewer chemicals, and all of the benefits that come from saving the environment, using less land, as well as not guaranteeing, but ensuring that the grower will have something at the end of the growing season. Because we know our growers should stay in business, because if there are no farmers, there's no food.
Exactly. We need it. We need all this kind of research to make sure we're going to have food in the next years. If some of the people that are watching us probably are interested in start vanilla produce also, or they have some kind of plants here, they can ask you, for example, at the university to run this genomics research of some of the plants they have?
So we can certainly help with the cultural practices. We have an article that's on the previous slide where people can. We do have a growing guide that's freely available in English, French, and Spanish. The DNA testing does have costs associated with it, and that's the biggest problem. I would love to genotype everything, but we have to find the funding to do that. So if there are a number of growers that want to get together to do a project together, and we want to bring in a university from Columbia, maybe we'll find a company that wants to start growing vanilla, but they want to test the different plants. We can certainly help with the analyses, but we need to find a way to pay for the sequencing costs.
So, Mr. Alan Chambers, University of Florida, thanks for your presentation. It's a really good opportunity to us to have this knowledge.